Thank you so much. And first, I would like to thank the ISAB for giving me this opportunity to present my work. So, we have done a very comprehensive analysis on the peripheral blood of for the type 1 diabetes samples, and what I'm presenting here is the highlight of the results. So just to give you the brief introduction about how the type 1 diabetes happens. So first you have a genetic risk, which is based on the HLAs, and then there is a beta cell stress in your pancreas, which leads to presentation of one or more autoantibodies. And at that stage, you already have a type 1 diabetes. But as it progresses, you have more than two autoantibodies, then you become more dysglycemic, and then you have a long-standing type 1 diabetes. How TCRs can help in this? So TCRs are basically the librarian which keeps the records of all the infection that you had in your lifetime. And we can use these records to figure out to either classify the disease in healthy or to find the specific antigen epitopes. What we have here, we have more than 2,000 TCR repertoires, and which is divided in 3 different cohorts. And the first cohort, we have all 4 different types. So we have type 1 diabetic patients. They're first degree relatives, which also has this susceptibility based on their HLAs. And then we have second degree relatives. They have less susceptibility and then controls. The cohort 2 and 3 are different they're sequenced with a different chemistry, but they contain the type 1 diabetic and control patients. And the goal we have, I will talk about these goals one by one. So let's start with the first goal. First, we want to see on the whole repertoire level, do we see any difference between the clinical groups? So what we found, we used 3 different matrices. So we used 3 different approaches to find similarity and differences. So we used Hill-based diversity profile. We looked at the overlap of the public clones and also the Wurster-Hahn index. And based on these 3 parameters, we found that there is no clustering, as you can see from these lines here. It doesn't cluster based on age, disease status, or their HLA types. So we concluded that the whole repertoire level, you cannot really find the difference between the control and type 1 diabetic. So we went further, more deep into it, and first we tried to look at how your HLA affects your TCR repertoires. So there was another paper which very nicely explained that HLAs really affect your TCR repertoires. And this is one of the example structure here. You can see the HLAs are in a very close proximity to TCRs, and there is an antigen in between. So there must be definitely some effect of these HLAs on the TCRs. And in type 1 diabetes, there are certain HLA types that are very high risk, and these are DR3 and DR4. To look into it, what we did is we looked variation at each site in HLA and each position in the CDR3 sequences, and we calculated the multivariate multiple linear regression model, which is basically tells like how much effect of the HLA variation is showing the variation in the different effect on the CDR3 frequencies. So we have done total of 27,000 tests, and based on that, we found only 47% of them had the significance. And you can see mainly the DQA, DQB, and DRB1 were the main where you found the most difference. So these are class 2 HLA types. So next what we did is we removed the HLA variation, and instead of that, we used HLA risk score, which is a previous study in which they have calculated the log-odd ratio of type 1 diabetes risk just based on the 3 HLA positions as described here. And based on that, we calculated the linear regression. We calculated the linear regression for each position and CDR sequence with the HLA risk score, and we found all these CDR3 phenotypes for different length of CDR3. You can see from the length 12 to 18. We found that these are positively or negatively associated with the type for the HLA risk, and we called them CDR3 phenotypes. And now once we have these CDR3 phenotypes, how can we use them to predict the type 1 diabetes risk solely from the TCR repertoires? So first we calculated the repertoire level CDR3 risk score, which is basically the sum of your CDR3 phenotypes and their effect sizes for the whole repertoire, and you average it. And you see that type 1 diabetes always shows a higher CDR3 risk score based on the repertoire compared to the FDR, SDR, and control. And they also show the trend that FDR has a bit higher than the SDR and then control. And this is the repertoire level. The previous one was the repertoire level score, but now we also went to the subsequence level, HLA motifs. So based on these CDR3 phenotypes, we tried to assemble them together for different length, and we obtained a motif. And this motif again showed that type and for the positively associated motif, you see that T1D risk score is much higher, and then control has lower. But when you see the NHLA motif score, which is the negatively associated, so these are the protective motifs, so they have much higher risk score for the control compared to the type 1 diabetes. So now we want to use different machine learning methods and see how they perform compared to this HLA associated risk score. So we use our HLA risk score as a baseline model, and the TCR features are just discussed as a baseline. So obviously the HLA risk score has the highest AUC, and then the CDRT risk score that we calculated on cohort 1 and cohort 2, 3, you see the performance is a bit lower than the HLA risk score, which is expected because we calculated all these information from the HLA risk score. But it is still good enough. Then we wanted to see, without using any HLA information, can we predict same thing with the TCR repertoires? And for that, we use different approaches, but I'm mainly talking about the deep RC here. But we have also used the information based on the public clones and also the KMR frequencies, but they had their own limitations. But deep RC is a multi-instance learning-based, modern half-field network-based method for classification. And when we use the deep RC, we see the performance is better than the TCR features that we just discussed. And it shows, again, much higher score for the type 1 diabetic repertoires compared to the control. But one of the main features of the deep RC is that it can also get the attention layer and try to get the sequences which has the highest attention in the machine learning, in the deep learning network. And we extracted these sequences and calculated a motif out of it. So it's a much more complex motif. As you can see, it has gaps in between, and which is not possible with the conventional shallow machine learning methods. And again, these motifs showed a good performance. It's a bit less than the whole deep RC model because it's a simplified version of it. But again, it has a pretty good performance. The main output from this deep RC is that when we stratified all these repertoires based on their HLA types, so I told like the conventional high-risk HLA alleles are the DR3 and DR4. So when the repertoire doesn't have these high-risk HLA alleles, so DRX by X and DRX by X doesn't have a high-risk allele, there you can see that deep RC was able to predict that which repertoire has higher risk or has higher risk for type 1 diabetes. And same goes for the autoantibody counts. So if the individual doesn't have any autoantibody present, it can still tell that which patient has higher risk for the type 1 diabetes. So this was a really good result that we obtained for the deep RC and was also validated with our TCR features. Further, we also tested these motifs on our spleen and pancreatic cohorts, and we also observed the same results there. I'm not showing those results there, but that was the conclusion, which is kind of a big results in the type 1 diabetes domain because none of the motifs were actually found in the spleen, at least from the peripheral blood. Overall, the conclusion of these reports is that we found that repertoire level similarity and diversity analysis cannot sufficiently classify type 1 diabetes status. And we know that CDR3 sequences are affected by the HLA alleles, and then we use this information to calculate the CDR3 phenotypes, and which can be used as a biomarker for that HLA association type 1 diabetes risk. And then we also trained our machine learning models without using the HLA information, and we found a pretty good performance as comparable to the TCR features that we obtained from the HLA. And then again, the T1D associated motif that we obtained from the deep RC, they were also able to predict for the repertoires which were not considered at high risk based on autoimmunity or your HLA risk. Thank you.
Thank you very much, Puneet. Yes, questions? Thank you. Thank you. Congratulations. I have a question, and is: did you analyze only HLA type 1, or did you analyze both classes?
We have tried both, and we found most association in class 2. Although in type 1 diabetes field, we found most association in class 2, but I know that in type 1 diabetes, most of the analysis are done on class 1 because it's much easier to analyze. But we also did TCR clustering, in which we found some association, the whole sequence association with the type 1, class 1 actually, yes.
Nice presentation. And the risk you were showing, they were related to DR3 and DR4,
Yes.
with the non-optimal stratification, because In principle, I think this is the Q2 and Q8 which are thought to be implicated, although they are in the 100 leg. Have you also done this analysis stratified by Q2 and Q8?
So when I talk about the HLA score, it is basically not based on the alleles, it's based on the variation in sites. So it basically is much more comprehensive than just saying DR3, DR4, DQ1, these things. So I think since our most of the list is based on the HLA score, which is more comprehensive, I think we have accounted for that.
So I have one question. Were you expecting differences when you were analyzing all tissues?
That would be the ideal condition, but that's very interesting that we found that. I have a slide here. I have just kept it hidden. So yeah, this is the where we have looked into the spleen CD8 cells and then in the tissues. Okay. Yeah. Yes. So these are the different variants they found from the genome-wide association studies. I cannot. Okay, these are the different risk alleles they found from there, and then this is the spleen CD8 where they found higher risk of diabetes based on these models, and this is the sorted cells. So it's kind of a very good validation on the results.
From the